Everyone. My name is Bruce Struminger, and I'm with Project ECHO at the University of New Mexico. And it's my pleasure to be the first to welcome you to today's COVID-19 vaccination capacity building for health workers learning session. Today's session is going to focus on understanding COVID-19 vaccines, safety, and efficacy. Next slide. We're just going to run through a few housekeeping items. First is that interpretation is available in French and Spanish by clicking on the globe interpretation button at the bottom right of your Zoom toolbar. Next slide. So you can see there English and French are available. Next slide. If you only want to hear the French or Spanish, click on mute original audio just below the English and French icons, and that should mute the English that would be in the background. Next slide. We're really looking forward to your questions and comments and going to be spending about 35 to 40 minutes focused on that. So please share those in the Q&A feature just to the left of that interpretation feature. If you have any IT or logistic issues or questions about the certificate of attendance, please put those in the chat and we'll be monitoring that as well and try to help you out as best we can. Next slide. This session is being recorded and your attendance is consent to be recorded. The recordings will be shared after the session in English, French, and Spanish, and we'll share a link to the website where you can find the recordings for today's session as well as past sessions. We're also offering a certificate of attendance through the University of New Mexico where Project ECHO is based. We'll share a link in the chat about 15 minutes before the end of the session. And we really look forward to your feedback. It'll take 2 or 3 minutes to complete a brief survey, and then you'll be able to get the certificate of attendance instantly and electronically. Next slide. Just a note about data use. We take data stewardship very seriously, and your individual data is going to be de-identified and kept confidential. What is shared to make maps, etc., is all de-identified. Next slide. Just to note that this live learning session is based on and complementing the Open WHO trainings. There are 2 training series, one for national deployment and vaccination planning, and another related to vaccination training for health workers. Today's session is complementing that series, and we'll share with you links to those online courses so that you can take advantage of them. Next slide. With that, it's my pleasure to hand the baton to Shoshana Golden at WHO. Shoshana.
Thank you so much, Bruce. And welcome everyone to today's session. Next slide, please. As always, we have a few announcements for you, and then we'll be diving into the session content. A couple of quick announcements. WHO has now listed the following COVID-19 vaccines for emergency use and provided recommendations for their use. These include AstraZeneca, both the SII and SK, Johnson, which recommendations will be published for this week, Moderna, and Pfizer, BioNTech, Comirnaty vaccine. To help you in actually using these vaccines, WHO is preparing short instructional videos on how to handle, prepare, and administer these specific vaccine products. You can find this information on the Open WHO COVID-19 Vaccine Specific Resources course as they become available. Currently, the Pfizer video is available and content is available for the 3 vaccines in blue. We will have videos available on this website probably by the end of this week or early next week for the majority of the vaccines I've just mentioned. Next slide, please. Today, we'll now be focusing on COVID-19 vaccine safety and efficacy. We'll start with a presentation from Madhav on what to expect from COVID-19 vaccines, followed by our what's next of upcoming sessions. And then the majority of this session, as Bruce mentioned, will be focused on your questions and answers. So please do use that Q&A feature to share your questions. Next slide, please. The goal for today's session is to share key information on how COVID-19 vaccines work. We have two learning objectives. The first is to increase understanding of how COVID-19 vaccines were developed and assessed. The second learning objective is to highlight key messages on safety and efficacy of COVID-19 vaccines. Next slide, please. I'd now like to turn over to Madhav, who will be giving us a presentation on what to expect from COVID-19 vaccines.
Thank you, Soshana. Can you hear me? Wonderful. Hello, everyone. I'm Madhav. I'm working in the WHO headquarters in the vaccine safety team. I'm now going to be talking on a very, very basic level on what to expect from COVID-19 vaccines, particularly from the context of the safety aspects. Because in fact, we want to keep this discussion very small and also the introduction very small, because we would really like to hear from you and also clarify your questions. And that's what the focus of this discussion, of this talk is going to be. And most of the questions we hope we will be able to answer in the Q&A session. So this is just a quick introduction. So next slide, please. So I'm just going to go to the basics, like, you know, so when you, one sec, I'm not able to get out. Can you give me a bigger screen, please? I cannot read this on the screen.
Madhav, you can use the Zoom feature at the top of your screen. So if you go to the top of your screen where it says view options, you can change your Zoom ratio to be 150.
Oh, okay, got it. No, but I'm not able to hide. Okay, got it. Excellent. Thank you very much. Thanks, and I'm sorry for that interruption. Thank you very much. So first of all, let us just start off with the basics. So when you look at the COVID vaccine, let us see about how to make a COVID vaccine. Now there are three main approaches to make the COVID vaccine. One is any vaccine for that matter. One is using the whole virus or the bacteria or the organism itself. The second one is actually certain parts of that organism which can actually trigger the immune system, or just the contents of the organism, which is actually the genetic material. Next, please. So when you look at the whole organism, supposing we use the whole virus or the bacteria, the way in which we can get it will be actually three. One is we can inactivate the virus by using certain chemical substances, like for instance formaldehyde, and you get an inactivated vaccine. Then the second one is actually a live attenuated vaccine, where you don't kill the virus itself, but you kind of keep it alive, and then you make it lose its virulence. And then what the third one is actually a viral vector vaccine, where the component, the inner component is actually replaced with something else, and then you get a viral vector vaccine. Now the next one, please. So the parts which, when you look at the subunit approach, you don't use the full virus, but you use certain parts of that particular virus or a bacteria, for which that is enough, because to create an immune response from the human body. Next one, please. Now, if you look at the genetic material, what we do is there are two different components of the genetic material. One is actually the DNA itself, the deoxyribose nucleic acid is used, or if it is certain parts of it is transcribed, and you can actually get just the messenger RNA to actually make the COVID vaccine. Next slide, please. So what is so different compared to conventional vaccines, and in terms of safety, why is this important? Now, when you look at the conventional vaccine development, normally vaccine development can take somewhere from about 10 years, 8 years, 10 years, and even up to 20 years. In fact, the shortest vaccine before the COVID vaccine, when it was developed, was actually the mumps vaccine, which was developed within a record period of 4 years. Whereas if you look at certain vaccines, even after about 30 to 40 years, we still don't have a perfect antigen. So when vaccines are being made, but when you look at the COVID vaccine, you must have realized that the COVID vaccine actually came into existence with much less than a year since the clinical trial started. So there has been a little bit of shortening of the clinical trial phases, and therefore there has been certain things why we should be a little bit more alert to the safety aspects. It's important. Now, when you look at the vaccine manufacturing, first of all, there is a preclinical phases. The first thing what you see in those colored circles there is actually, identifying the molecule, which actually helps you to identify the vaccine, to identify the component which can trigger an immune response. And then this is then used in certain animals for clinical trials. Basically, they look at two things. One is whether it is safe, and the second thing is whether it is effective. So they do testing on mice, sometimes hamsters, sometimes they also do it on primates like, monkeys. So they test it and they try to find out. Now, once it is found to be safe and effective in animal models, that is when you have the clinical trials which actually start. Now, there are usually 3 clinical trial phases. The phase 1 is actually tested usually in healthy people, approximately in small number of volunteers. You can have something like 40, sometimes up to 100 people who get who actually are tested. So this is basically the phase 1 clinical trial testing. In phase 2, it is usually done in healthy adults. In phase 2 clinical trials, they increase the sample size, and then from hundreds it goes up to several thousands. From hundreds it can go up to 1,000 or even several few thousands. It can go up to testing in phase 2 clinical trials. So this is when basically what happens is they test them on healthy target population. So they like to test it on the entire spectrum of the population which the vaccine is going to be given. Again, they look at 2 things. One, they like to know the dose. This is where it's calibrated. They like to know whether the dose, which is the best optimal dose, they like to find out. They also like to find out about the safety patterns as well as the efficacy patterns. Then it goes into phase 3 clinical trials where you have people with other comorbidities and other conditions which also get added up. And here they do it on several thousands of people. In fact, if you look at vaccines like AstraZeneca, you have something like around 60,000 people who are actually tested in phase 3. Now, after it is in phase 3, vaccines are licensed, and then it starts coming into, it is in case of COVID vaccines, it is released under what we call as the emergency use authorization. And then we start off with the mass immunization for the public, for everyone. Next slide, please.
Madhav, can I please remind you to speak slowly for the interpreters?
Oh, sorry, thanks. Okay, so now let us see what to expect from a COVID vaccine. Now, ideally, a vaccine, if you see, the best vaccine is one which is able to prevent infection. Now, so on the extreme left, what you see, it is actually meant to prevent infection. Now, what is the meaning of the word prevent infection? It should ideally prevent the COVID virus from even, as soon as the virus enters into the human body, it should be able to get inactivated. But then right now, none of the vaccines which we are having is able to produce that. So the next best thing would be, so in other words, it is able to even prevent, the best vaccine will even be able to prevent the asymptomatic infections. So, but as I said just now, this is not possible. At present, none of the present vaccines are able to do that. But then, even if it doesn't prevent asymptomatic infection, it should be able to prevent disease. Okay, fine. If it prevents disease, then better, some of them may not prevent the disease, but it will at least prevent severe disease, so it prevents hospitalization. So you may have something like a moderate disease, but you may not get into severe disease. Now, even if you get into severe disease, you may not actually prevent, some of the vaccines prevent very severe disease, it prevents you from going from severe disease to critical disease. It prevents that. And some of them actually prevent from critical disease to death, so this is what it is. And also, vaccines are expected to prevent transmission of the disease into the community. Now, so when you look at all the vaccines which have been manufactured right now, most of them, almost all of them, are actually effective against severe disease, and also they are able to prevent death. But if you see, many of them may not be able to prevent the virus from entering into the human body and multiplying. Next slide, please. Now let's look at it in terms of the vaccine platforms which we are having. Actually, even though this looks like a little bit of a complex diagram, it is not very complex actually to say. I would like you to focus first on the virus itself. Like if you see the central picture, the circle with the spikes which are there, that is actually the central virus which is present. Now in the virus, you will see some black color spikes which is actually coming out. Now, the wax, it has got two parts, this is that what you see in black is actually called the spike proteins. And what you see as the tip of the flower is the one on the tip of each of the spike protein is the place where the virus attaches itself to the host cell and where it tries to enter into the host cell. Now, the vaccines which we are manufacturing, particularly the vaccines which are available right now, the RNA vaccines, as well as the viral vector vaccines, they are able to produce antibodies against the spike proteins, and then it is able to prevent the disease. So if you see, so this is in a nutshell about how the vaccine actually works at present. Of course, you are also having certain vaccines which are actually the killed vaccines, which where the whole organism itself is supposed to be producing the immune mechanism. So if you just, can you click once more, please? If you just click next, please. So if you next, there is supposed to be an animation here. Yeah, correct. So if you just look at this particular particular options which are available as vaccine platforms now, the ones on the top, that is basically the immune cell therapy, is not very commonly used. But then we are beginning, and also similarly protein and peptide subunit vaccines are not very commonly used. But you are already having something like inactivated vaccines are already available, and also attenuated vaccines are also being manufactured right now. Now, if you see, the ones which are present below are the ones which are going and which are very unique. That is, the one which you see on the right side is actually the viral vector vaccines. And I think I told you what a viral vector vaccine was in the beginning of the session. And what you see on the left side is actually what we call as the nucleic acid based vaccines. There are two different types of nucleic acid based vaccine. One is the mRNA vaccine, and the other one is your DNA vaccine. So this is basically the very unique type of vaccines which has come into existence after the COVID after COVID pandemic has started. Now you can ask me, is this anything new? When you look at the viral vector vaccines, the one you see on the right side, actually, no, it is not very new, because it has actually been used earlier for Ebola. And so it is not really very new. Whereas if you look at the nucleic acid vaccines, the one you see on the left, is it new? Yes, the technology is a little new, but it has still been used originally for the treatment of cancers. And it has also been used extensively in veterinary use for quite a long time. Next slide, please. So I'm not going into very much complicated details here, but I would like to say that if you just see the nucleic acid vaccines, as I said, what you see on the, let us first go through the left side of the screen, and then on the white part, if you see, there is a DNA vaccine and then there is the RNA vaccine. Right now, there is no DNA vaccines which are yet manufactured. So let us look only at the RNA vaccines. So what is the RNA vaccines which are available? They are actually the two vaccines, which are actually the Pfizer vaccine as well as the Moderna vaccine. So what does it contain? If you just see, what you see in the form of that blue dotted circle around is actually the lipoprotein. It is actually a lipoprotein envelope inside which there is a messenger RNA which is actually put inside. Now you can ask me, why is that? It is very simple, because the messenger RNA by itself cannot be injected into the human body because it will get destroyed. So because of your enzymes in the human body. So the lipoprotein envelope which is present there on the top will protect it. It comes and it attaches itself. Once you give that injection and once it is given into your deltoid, it will come into the tissues next to that. And when it enters into the cells within that particular tissues, the lipoprotein envelope attaches itself to the cells, and then the messenger RNA enters into the cytoplasm. And then there it will actually go into a particular part of the cell called the ribosomes, where it will program the cells to create the spike proteins. So as you see, the viral spike proteins will then come outside, and then the body's immune mechanism identifies the spike proteins. And once the spike protein gets identified, the body will start creating antibodies. And these antibodies, when it identifies a real coronavirus at a later date, it will mount an immune response. So this is the way in which this is acting. Now, so when you look at this, this particular thing, let us go to the green part of the slide. It is actually if you just see, it is a lipid nanoparticle, and it is highly immunogenic. And one of the great advantages, particularly of the RNA vaccine is the fact that it can be rapidly developed. It is not very expensive. And not only that, with this antigenic variants which are coming up, you can actually tweak it. It can be adjusted to make newer varieties without much of a high level technological changes. But then the biggest problem is that, you know, sometimes you can get adverse event depending on the sequence of the genes which are actually introduced inside the RNA. It can actually be there. And then there is also a theoretical risk. You must have heard about conditions like antibody mediated immunity. So those kind of conditions can also sometimes come up as a result of this vaccine being administered. And when DNA vaccines are, as I said, not available yet right now, but then there is a theoretical risk about that the DNA can enter into the host cells. But again, this has not been proved so far in animal or human studies. Next slide, please. Now let us go to the viral vector vaccines. Again, there are two types. One viral vector vaccine is the one which we call as replicating, and the second one is actually what we call as non-replicating. As the name indicates, what happens is we take the capsule of one particular virus. Like if you look at the AstraZeneca vaccine, they use something called the chimpanzee adenovirus. So that is going to be the capsule which is taken, the shell which is actually taken, and inside that there is actually the genetic material introduced. The coronavirus spike gene is introduced into that, and when the coronavirus spike gene is introduced into that, it is then once it is injected into the human body, this gets expressed, as I just mentioned right now, and it stimulates the body to produce the spike proteins. I wanted to also alert you that the AstraZeneca vaccine is a non-replicating viral vector vaccine. Okay, so as I just mentioned, the advantage, let's go to the green part. The great advantage is the fact that it results in cell-mediated immunity, but then the disadvantage is that, you know, repeated doses may induce immunity to the viral vector. Sometimes you may have to, so the single dose or with different vectors. So this is a bit of a problem. And also it depends on the source of the virus. The risk depends on the source of the virus, the pre-existing, supposing if the body itself has got some protection again, has got some immunity against the virus, I mean the shell which is getting injected, then it can actually have a bit of a problem. And sometimes there is also the last bullet, what you see there is a theoretical risk of mutation. So this is also there, but it has never been proved. Okay, it has never happened in real life. Next slide, please. So let us quickly look at it from the safety perspective. And when you look at it from the safety perspective, what we know and what we don't know. When you look at the viral vector vaccines, when we look at the vaccine platforms, we know that these vaccines are quite effective based on your clinical trials. And also we notice that in places where vaccines are given in the world, the incidence of this disease is actually coming down. And then so we know that it is actually quite effective. But then we also do not know how long is this effectiveness, how long is it effective? Do you need to give booster doses? These kind of things we really do not know. Now, when you look at it in terms of manufacture, you know, when you look at the manufacturing, when the manufacturing occurs in a small scale, particularly for clinical trials, we know that it is safe. And then basically when smaller doses are given, it is quite okay. But when you manufacture it in bulk, you know, for instance, millions of doses are being made every day. And when you start administering it in bulk, we really don't know whether what you're seeing on the clinical trial in a small scale can actually be replicated in a bigger scale. Then the next thing is with regard to the transport and storage. Now, especially when you look at your mRNA vaccines, we need to have cold temperatures. Like, you know, you must have heard about -70 for the Pfizer and -20 for the Moderna vaccine. So this is something which is sometimes very challenging, particularly in low and middle income country settings. So that is one problem. The second thing is we don't know what we don't know is because of this particular storage temperatures. If something changes from the standard temperature, how is it going to affect the vaccine's effectiveness and safety pattern of it? Now, the outcome of vaccination, it is used to prevent disease. But that, of course, we know. And all the vaccines, as I mentioned in the beginning, prevent severe disease. But as I said, we don't know to what extent it prevents infection, prevention of severe disease, and its effect on population transmission. So when you look at coming to the adverse reactions, people are very much aware of short-term reactogenicity. For instance, you will always find people who always come back and then vaccinate today and then immediately you 1 minute. So this is so you get people you get a program where they say that the vaccines are today we conducted so many cases, so many patients, and so many persons, and after that there has been no problems. But we really don't know what will happen in the long-term effects, like what will happen after a week, after a month, after 6 months. That is number one. And one more thing what we don't know is how will the vaccines behave in subpopulations? How will it behave for people with pre-existing morbidities, like cardiac conditions, your hypertension, diabetes, ischemic. So those kind of conditions we really do not know. And the efficacy and safety we know is very good in less than 65 years. But also the level, the clinical trials yesterday, the one which just shown now, the Johnson & Johnson vaccine, we know that they have done some clinical trials for above 65 years. But many of the other vaccines, the clinical trials were like between 18 years and 60 years. Then it's also in certain unique populations like pregnancy and also among immunocompromised, we are not really very aware of the safety profile. Next slide, please. Very good. So this one, this is quite an important slide, and I purposely put it up from the safety perspective to give you an idea on what happens. Now, I want to draw your attention into 3 parts in it. If you just go to the top, this slide is actually describing how does safety data. You are a health worker. You're sitting in the periphery. You have vaccinated a particular person and the person develops an adverse event. Now, what has happened is you are reporting this particular adverse event and you really don't know how does your, that small bit of information you provide, how is it actually transmitted to the global level and how is it being used? So I'm actually going to, this slide has got 3 parts. On the left side, you have a subnational part. In the middle, you have a national part. And on the right side, you have a global part. So if you just look at the subnational part, let us go right to the bottom. Now, right at the bottom of that, you will see 3 people sitting there in the bottom. One is the health worker, the second one is a nurse, and the third one is a doctor. Now, after a person is vaccinated, the person might develop an adverse event, and then the person comes to one of these people and then says, see, I have a particular problem. And when that happens, what happens? The reporting form, there is a small, every country has got a small standard AEFI reporting form. Now, this is a single page document. It is actually filled up by this particular group and one of these persons, and it goes to the district, what you see in the middle. Now, some of the countries have got electronic methods for collecting data. Sometimes, the data entry can take place in an electronic method, and then it reaches the district level. Now, from the district level, there is usually something known as data cleaning. So they look at that particular information you are providing as health staff, and at the district level, what happens is they filter this particular data and divide it into 2 categories. One is serious categories and non-serious categories. Now, the serious categories are conditions like, death, hospitalization, disability, congenital anomaly, and if there is any other event of medical interest. So this is actually coming up in the middle. Now, once if it is a serious adverse event, that means like if there is a death or a hospitalization, an investigation, a detailed investigation is initiated from the district level. So that is what happens. Now, what happens to the data, what you are sending in the reporting form to the district? It is actually sent from there to the province, or in some places it is called the zone or the state, if you go upwards. And in the zone or the province or the state, there are 3 groups of people who handle the data. One is the immunization program itself, who are living in the province, who are actually in charge of the whole province. The regulators who are present in the province, and then the disease surveillance people. So these people are also sharing the data among themselves, and then they transmit the data to the people in the middle column. That is, they put the data into the national database. And the national database, what you see in the middle column in the form of the hard disk, the disk which you see, the database there, actually shares the data between the EPI program as well as the regulators. So there is a huge amount of sharing of data which takes place between the regulators as well as with the immunization program. The manufacturers, the vaccine manufacturers also share important information to the national regulatory authority. Now the data, the national data, which is shared between the EPI and the regulators, are then transmitted, are actually converted. Just like, when you talk about data being shared with someone else, you need to talk about data confidentiality. So what happens is the data is encrypted into a particular format known as the E2B format. And then it is shared into the global database. What you see on the right side at the bottom, the picture, what you see there, it is called the WHO Program for International Drug Monitoring. There it is, all the countries in the world put the data into the global database. And then from there, the countries then share the data, once it is put there, the information is collected by WHO and also by two teams. One is the pharmacovigilance team, and then also the pre-qualification team. So these two teams actually are responsible for the data collection, for processing. The pre-qualification team also gets information from the manufacturers about periodic safety. What are the, they give something called the periodic safety update reports. So this is also shared with WHO. And then from WHO shares it with committees. You must have heard about, the SAGE, the Strategic Advisory Group of Experts. So they are able to share it with those committees, as well as with the Global Advisory Committee on Vaccine Safety. Next slide, please.
Manav, I'm sharing the 1 minute mark. We'll need to
No,
then
it's over.
transition.
It's finished. Just one more slide. That's it. Finished. Okay. Thank you very much. And so this is actually, since we are running short of time, what you see on the screen, these are the as of the 10th of March, which is the pre-qualification, the vaccines which have actually gone through the WHO pre-qualification process. As mentioned, we just yesterday only the Johnson & Johnson vaccine was actually cleared through the SAGE. Next slide, please. So this is the last slide. And this is when the slide set is being shared with you, you are most welcome to share the links which are here, which will be able to provide you with more information. Back to you, Soshana. Thank you.
Thank you so much, Madhav, for that fantastic overview of how COVID-19 vaccines work. That was really wonderful. Next slide, please. We'll briefly talk about what's next with the ECHO seminar series and then turn to you for your questions. So next slide, please. As always, recordings from this session will be available in English, French, and Spanish. You can continue to ask questions and share your experiences after the session on TechNet and/or on Telegram, as you prefer. Upcoming topics for next Tuesday will be generating acceptance and demand for COVID-19 vaccines. This is intended for our national and subnational focal points, and the registration link is available here. For health workers, our next session will be on Tuesday, 30th March, focused on reporting on COVID-19 vaccines, both monitoring and IEFI. On 6th of April, again for our national and subnational focal points, we'll be talking about vaccination strategies for COVID-19 vaccine. The slides from today's session will be shared out after the call. Next slide, please. We'll now be turning to our question and answer portion of today's call. For this, what we'll be doing is collecting your questions from the Q&A feature, so please feel free to include as many questions as you'd like. Those that we answer now, we'll try and do both in written and in verbal format, and any unanswered questions related to the topic, we will share with our technical experts after the call for them also to respond in a written form. With this, I'd like to ask Madhav, Melanie, and Alia to please turn on their videos, and we'll start looking at our Q&As. The first question for Madhav and for Melanie is there's a number of questions about long-term side effects for COVID-19 vaccines and short-term, what are the major side effects for different types of COVID-19 vaccines. I know, Madhav, you briefly touched on this in your presentation, but it would be great if you guys could give a brief explanation for the short-term and long-term side effects that health workers can anticipate.
Okay, let me just quickly start off. First of all, we should be very clear to understand what we mean by this short-term and also long-term side effects and what we know and what we don't know. Now, as I mentioned to you before, at the time of there are phase 1, phase 2, and phase 3 clinical trials, and during this particular phases, you know what they do? They vaccinate these the people for who receive the clinical trial participants, and they carefully follow them up. And all the information which we are having currently is based on the findings of those clinical trials. That is one. The second way in which we get the information is based on the reports which health workers like you provide to the levels which I just showed you. That is actually what we call as spontaneous reporting. Now, let us based on the clinical trials. What do we have? Now, based on the clinical trials, you're basically dividing the adverse events into two broad categories. What are they? One is the local site reactions. That means what happens at the site of injection. So usually if you see, there is a lot of problems which you can find, and you will find pain at injection site, swelling, and some people sometimes get even a small rash around the site of injection. Those kind of things are very, very common. So please expect that. And you need to tell health workers that this is what you have to expect. Now, so that is number one. The second thing which you should also expect is something called systemic. Now, this again, if you see the systemic adverse events which occurs is conditions like, for instance, fever after this thing. Some people develop nausea. Some people develop rash. Some people develop very, very high fever. Some people develop even lymph node enlargement. Now, so that is one more thing which you have to keep in mind. So this is the immediate short-term adverse events. Now, that said, there's another thing which you have to keep in mind, and that is actually the long-term adverse events. Now, when you look at it in terms of long-term adverse events, from the COVID vaccine perspective, right now, other than probably lymphadenopathy, we really don't have much information as to what could be. And that also, again, short lasting for about less than a week, for instance. We don't have any information as to long-term side effects. Even though, for instance, there are information which we have during clinical trials. What were the information we had during clinical trials? There was when the clinical trials, when you look at it, there are some information which we get called certain events of interest. This is called adverse events of special interest. Let me give you an example. For instance, when the Pfizer vaccine clinical trials were being conducted, they noticed that there was a little bit more Bell's palsy, that is the facial nerve paralysis, was occurring much more in certain groups. But then when they had it with the entire population, they found out that it was not very significant. But just to be on the safer side, they said, we need to be carefully following up these events of special interest. So please note that, as far as we are concerned, when short-term side effects, we are very much aware of, but with regard to long-term side effects, we are still in the process of looking for it. Over.
Thank you so much, Mana. We now have a number of questions on the AstraZeneca vaccine and the reports of blood coagulation issues. I'll turn again to our panelists to respond to this question.
Melanie, you want to go first?
Madhav, would you like to begin?
No, because I'm doing all the talking, so I was just wondering whether someone else wants to take it up. I'm happy to, or I can do it. Yeah, okay. So, fine. I think I should be taking up this question as well. So basically, you must have all read that originally it basically started in Austria last Sunday, not this Sunday, the previous Sunday, that there were a couple of reports of coagulation disorders. What were the coagulation disorders reported from Austria? there was one person who had developed some kind of pulmonary, basically clots, pulmonary embolism, and also there was another person who had also developed cerebral embolism, and it healthcare workers, and that is the reason why there was a little bit of a worry on that particular issue. So what happened was, as a precaution, many of the countries actually stopped the, initially they thought that it is connected to one particular batch. And after seeing this, some other countries in Europe, exclusively in Europe primarily, and also in Thailand, what they did was they said that they will temporarily halt the vaccination, pause the vaccinations until they get additional information. Now it is actually like yesterday, over this current weekend, we noticed that there were information from big countries like France, Germany, from Italy. So these countries had also mentioned that they are finding out some amount of concerns. As of today as we speak, the information is that this is still being debated, and today afternoon there is going to be a global advisory committee meeting on vaccine safety, where this will be kind of debated by using data, and based on that particular data, WHO will come out with a particular position. Over.
Wonderful. Thank you so much, Mada, for that helpful response. For Alia, we're receiving a question about the no-fault compensation mechanism. Can you guide folks to where they can learn more about the NFC?
Yes, absolutely. I've put the website on a couple of the answers because there were two questions. So I've put them as answers. So it's covaxclaims.com is where you will get the information. So basically you will need to know, so this program was set up for the advanced market commitment economies. That's 92 economies that get vaccine through the COVAX. And it provides a sort of a fair, no-fault compensation mechanism for anybody who has permanent, serious adverse events or death. And basically, I mean, there's a lot of information there. The program is, the website is going live on the 31st of March. However, you can already access it and get a lot of information like how to submit an application, who's eligible, Q's and A's. Some countries have already received the vaccines and started vaccination. So if any eligible candidates actually has a serious adverse events, permanent adverse events or death, before the program goes live, they can still apply because there's quite a large window of opportunity for submitting applications. Happy to address further questions.
Thank you so much, Alia. For Melanie, we've received a couple of questions about how COVID-19 vaccination be conducted. So this is specifically for whether or not post-COVID-19 patients should be vaccinated, and whether elderly, so people above the age of 65 years old, should be vaccinated. Can you please comment on these two?
Sure, happy to comment on that. So on the aspect of vaccinating after a confirmed SARS-CoV-2 infection, currently what WHO recommends for the vaccines that they have issued recommendations on is not to vaccinate in the acute phase of the illness, but patients may be vaccinated after that acute phase has terminated. That said, individuals may, because there is a certain level of protection conferred by infection, currently data suggests it's up to 6 months of protection. So individuals may decide to defer vaccination until those 6 months have passed. But it's an individual decision, and there's no reason to not vaccinate once the acute illness is over. On the aspect of vaccinating of older individuals, to date on the vaccines, again, where WHO has issued recommendations, all of them have no upper age limit, i.e. they can be vaccinated starting from the age of 16 or 18 respectively without an upper age limit.
Wonderful, thank you so much, Melanie. And we have a follow-on question which is, what is the time period that health workers should observe between COVID-19 vaccination and other vaccinations? For example, the influenza vaccine.
So currently WHO recommends an interval of 14 days between COVID vaccination and any other vaccines.
Wonderful, thank you so much. We now have a question on why the COVID-19 vaccines do not use a vaccine vial monitor, and how safe are these vaccines? Mara, could you please come in and answer this question?
For the vaccine vial monitors, I just wanted to tell you that, there's a huge amount of research which goes on with regard to vaccine vial monitors, because it has to be, from what I understand, has to be tailor-made to the vaccine. And since we are deploying it very rapidly, the time taken for that particular research, we have to test it on field conditions and all that. So it's likely to take some time for it to come. That's number one. Number two, we are kind of expecting the vaccines to be administered in a large scale very, very quickly. So at present, for at least for a short period of time, the vaccine vial monitors are not being used. But we hope that very quickly that this will be coming into use very quickly. What is the second question, Soshana?
Sure. So we've got another question about, let me see. You respond to this, Mara?
Yeah, that's so what happens is, what is the meaning of the word vector? So what they do is, basically you're having something like, when you look at your capsule, you take out the component of the capsule, you will see the central powder inside. So what is happening is, it is something which looks like that. So what the shell of the capsule, the gelatinous substance, is taken from one virus, that is the chimpanzee adenovirus. And the central component of that has been tweaked to introduce a genome from the spike protein of the coronavirus, so that it will be able to form a single capsule. So that is actually what we call as a viral vector vaccine. I hope I have explained that.
That's wonderful. Thank you so much. We have a question about delayed AEFI and the last possible time. Alia, I'll turn to you to talk about the NFC, the no-fault compensation mechanism, and delayed AEFIs. Alia, I think you're muted.
Sorry. So the question is, can you apply for the no-fault for compensation if you have a delayed adverse event?
Exactly, and the last possible time frame.
Yeah, so the time frame really depends on a number of factors. So applications can only be done after 30 days after the adverse events. You cannot apply, you need a 30-day period unless there is a death, obviously. That's really to ensure that it's a serious and permanent adverse injury that is caused by the vaccine. In terms of the timeline, so there's two components to it. The first is that you have a 24-month period from the time the vaccine has been put into any market. And from that date on, there is a 24-month period where applications can be made for that particular vaccine. And then once beyond that 24-month period, there's an additional 36 months period. So you have a maximum of about 5 years to submit an application in terms of timeline. There is no really timeline in terms of like how long after, but they assess, so you have the administrator, the claims administrator, they have review panels, and they assess based on, is this a most probable cause of the adverse event? So if it's later, it will still be assessed, and based on that, the claim would be either accepted or not.
Wonderful. Thank you so much, Alia.
Soshana, can I just come in, please, in addition to one more point which Alia brought in?
Absolutely.
Thanks, Alia. I just want, since this happens to be a target audience meant for the health staff, I want to also alert you that documentation is very, very important. particularly the name of the manufacturer. It is very important. The batch number, everything is so important. So please, if you're having an adverse event, I'm just alerting you that if the documentation is improper, then all what Alia said will not work. Over.
Absolutely. Thank you very much, Madhav. This is really critical to collect. And in fact, so there is two for the application process. There is one form that the applicant themselves or a legal representative of them fills out. And the second form is filled out by a healthcare worker. And this is where you really need the batch number, the manufacturer expiration date, all this critical information to be able to submit, to have a complete file for consideration. Thanks, Madhav.
Wonderful. Thank you, Madhav and Alia. Melanie, we have a question on why some COVID-19 vaccines should not be shaken. Can you please comment on this?
Sorry, should not be what?
Shaken. So some COVID-19 vaccines, the instructions are not to shake them, which is not typical practice for COVID-19, for vaccines in general. Can you comment on this?
Sorry, I may turn to Madhav. I'm not very firm on the handling of the vaccines. Thanks, Madhav.
Soshina, could you please repeat the question?
Yes, so the question is, some COVID-19 vaccines, the instruction is not to shake the vaccine, but instead to swirl the vaccine. Why is this?
Yes, okay. So now not only COVID-19 vaccines, right? It's even when you look at it in terms of measles vaccine and your regular vaccines, basically we find that you are expected, particularly when you're talking about reconstituting vaccines, where you need to talk about reconstitution, particularly when you talk about freeze-dried vaccines and all that. This is important. It's applicable not only to COVID vaccines, it's applicable to everything. You are not expected to, shake it violently and create a froth when you're actually doing when you're actually vaccinating. There are some very important reasons for that, because if you just look at it, the molecules need to be evenly spread out, but for that it is just enough to just tap it in the hand or gently swirl it around instead of trying to create more amount of bubbles to enter into it. That said, I also wanted to tell you something. When you look at it in terms of the mRNA vaccines, as I mentioned earlier, it is a suspension which actually includes the lipoprotein molecules. Do you remember? I just told you very clearly that the mRNA is located in between, and then the lipoprotein cover is actually present on each of that mRNA molecule. So this would, by violently shaking the contents, it could actually disrupt the general architecture and the molecular structure of the vaccine. Over.
Wonderful. Thank you so much, Madhav. We now have a number of questions on people who say that, who ask whether or not the COVID-19 vaccine will be protective in keeping people from transmitting the infection. I'll turn to Madhav and Melanie to comment on whether or not the COVID-19 vaccine is effective in preventing transmission to other people. Melanie, would you like to start?
I'm sure, happy to start. So, the short answer is we're still lacking data, and we're not sure yet whether the vaccine protects against transmission. This needs to be awaited, and it needs to be seen in larger phase rollout of the vaccines. There's some indication that some vaccines may prevent transmission, but we really don't have the data to be conclusive on that yet.
Thank you so much, Melanie. We have several questions about some of the specifics of COVID-19 vaccines. So Madhav, I'll share these with you. The first question is, what are the possible risks of integrating the COVID-19 viral DNA into the host cell DNA? A second question is, does the replicating properties of some mRNA vaccines determine the duration of immunity generated? Madhav, can you respond to these two questions?
Yes, I during my presentation in the beginning, I think I had made it very, very clear that at present, like if you just look at how does the mRNA actually work? The mRNA enters, see, you're beginning to ask me some technical questions. You enters the cytoplasm, and then from there it enters into the nucleus. And when it enters into the nucleus, it does not, I repeat, it does not integrate with the DNA which is present inside the nucleus. Instead, what it does is the cell is getting programmed to give instructions. There is something called the rough endoplasmic reticulum in the cytoplasm, and inside that there is this ribosomes. So what does it do? It gives instruction to the ribosomes to create copies. So it's like creating photocopies of the mRNA, which will then stimulate the creation of proteins, that is the spike proteins, and these spike proteins will be coming out of the cell, and then that spike protein which get exudated out of the cell, which actually will be seen or will be made visible to the immune system, which will mount first of all a T cell response, and then it will mount a B cell response, which will in then turn produce antibodies which will protect. But as I said just now, there is no integration of the mRNA into the host cell DNA. Over.
Thank you so much, Madhav. We have a question about what narrative can vaccinators use to help their patients understand efficacy and safety of the vaccines. Madhav and Melanie, would you like to comment on this? What should health workers be saying to their patients? I know that we do have a number of resources from our previous session, but wanted to open up this question to you as well, if there's particular resources that health workers should use.
Melanie, do you want to go first or shall I?
Please go ahead.
Okay, one of the most useful resources which I have found, by just as what Shoshana mentioned just now, for the COVID vaccine specific communications, there in that link what we gave you in earlier in that slide presentation, there is a particular link which clicks. If you click on that link, it opens out 9 modules, and one of those modules is specifically COVID vaccine specific. So you can communication specific, COVID vaccine communicate, because it has got 9 modules. It talks about adverse events following immunization, adverse events of special interest, so many things. So what happens is, since it basically talks about the communications related to COVID vaccines, that will be helpful. That's number 1. Number 2, I also would like to suggest to this particular group that there's a beautiful module which has been created by WHO Euro. It is actually the book is actually called Vaccine Safety Events, Managing the Communications Response. In fact, if I have some time, I can even share the link with you. It's one of the very simple book, very nicely written, and it also tells you in very, very simple, practical terms how to handle this kind of situations. Over.
Wonderful. Thank you so much, Madhav. And it's a great opportunity to promote that COVID-19 vaccination health worker training. Very much agreed. We have just 3 minutes left, so I'll ask just a couple more questions, and the remaining unanswered questions will be shared with our technical experts for their written comment and will be shared with all participants after the call. A couple of questions for Madhav and Melanie. Were any tests done for vaccine interactions with medications for other disorders, for example, epilepsy? Are there any studies done on the effects of AstraZeneca vaccine on immunocompromised individuals who are not on treatment? Can you please respond to these 2 questions about potential contraindications and the use of these vaccines?
Melanie, you want to go first?
Happy to start. So for the currently WHO recommended vaccines, there have been a certain number of people with comorbidities included. That varies by vaccine, but it's between 10 and 60% of people with comorbidities included. So we do have some data on people, for example, with diabetes, with cardiovascular disease, and other risk factors that may also increase the risk of severe COVID. So for those where there is data available, we do recommend the use of the vaccine in those specific groups. Where we do not recommend if the person has had a severe hypersensitivity reaction to a previous vaccine, so that would be a contraindication.
Wonderful, thank you, Melanie. I'll close us out with one last question. on whether there's an independent website to track COVID-19 vaccine adverse events globally. Madhav, can you comment on this?
Whether there is an independent?
Whether there's a website that tracks COVID-19
Oh, yeah,
vaccine
right.
adverse
Thanks.
events.
Thank you very much. I think this is a very, very important question. So who is actually monitoring this, this particular global level, at the global level? Now, you should understand that there is something, as I mentioned earlier, there is during my presentation where I showed you the flow of data. The data which all of you are, each health worker, that single piece of paper which you are putting, actually, do you know that the data travels all the way to the Uppsala Monitoring Center in Sweden? Now, the Uppsala Monitoring Center, which is located in Sweden, has got more than 20 million ICSRs, or the individual case safety reports, already since 1978 or something like that. So they are collecting adverse drug reactions, and also now they are also collecting adverse events following immunization. So this particular data, which is located in Sweden, who are the people who are able to access it? Within your countries, you have the National Regulatory Authority. The National Regulatory Authority in your country has got the full right to access this particular database. So what do? They will be given a username and password, and all from throughout the world, they will be able to have an idea. So this makes it all the more important, which I wanted to alert you, is to make that you collect the data and transmit the data to the global database. It's very, very important, because sharing of information only leads to action, because only then can we draw conclusions.
Wonderful. Thank you so much, Madhav. And thank you again to all of our speakers on today's call, Madhav, Melanie, and Alia. A big thanks as well to our fantastic interpreters, to the ECHO team, and to all of you for joining today's session. We will now conclude today's session. We'll be sharing the recordings in English, French, and Spanish after the call, and we'll be answering those remaining questions by a written form. With that, I would like to say, have a wonderful day, and give it over to our panelists for any closing words.
No, thank you very much. It has been very useful talking to all of you. And I think each and every one of you play a very, very important role in rolling out safe and effective vaccines. Over.
Wonderful. Melanie, Alia, any final parting words?
Nothing to add to what Madama said. Thank you very much for the invitation.
Wonderful. Alia, I think you're muted.
Sorry, that's the line. Just to thank everyone for their participation and their questions.
Wonderful. Thank you so much, and have a wonderful rest of your morning, afternoon, or evening.
Thank you, everyone. I'm just closing this meeting. Thank you so much.